Hi, everybody, and welcome to our module on exercise and starvation. Let's start this module by talking about the biochemistry of exercise. When you begin exercising, you rapidly deplete the ATP stores in your muscle cells. And if you want to be able to keep exercising, you need to derive energy from some other type of fuel. Now, there is glycogen stored in your muscle cells. There's also glucose available in the plasma. And these things can be fed into the TCA cycle to generate more ATP, but that is a slow process. So the body has evolved a mechanism in order to maintain maximal energy for a short period of time, and that involves using a substance called creatine as a fuel source. Creatine is present in your muscles and in your heart as phosphocreatine, and it serves as a source of phosphate groups. This is the structure of creatine on the left side of the screen here, and it can be phosphorylated by an enzyme called creatine kinase to form phosphocreatine, which has a phosphate group added to the structure. And you may be familiar with creatine kinase from studying cardiology. Elevated levels of creatine kinase are seen in patients who have a myocardial infarction. And the purpose of phosphocreatine inside your muscle cells is to donate this phosphate group to ADP to create molecules of ATP very rapidly without having to wait for glycolysis in the TCA cycle. So phosphocreatine serves as a reserve of phosphate groups for when ATP levels fall rapidly during exercise. You may be familiar with a substance called creatinine in the plasma, and you should know that creatinine is derived from creatine. Creatine undergoes spontaneous conversion to creatinine at a steady rate. The amount of creatinine that you find in your plasma is proportional to your muscle mass, which is where the creatine comes from. And then creatinine is excreted by the kidneys, and you may know that you can use it to estimate the glomerular filtration rate. Even though you have ATP in your muscles and you have creatinine around to donate phosphate groups, there's still a limited supply of these two substances available, and they're consumed within a few seconds of intense exercise. So their most important use is for short, intense exertion, like heavy lifting or sprinting. You may know that some bodybuilders consume supplements of creatine, and what they're hoping to do is increase the amount in their muscles so that they can exert themselves maximally for longer periods of time. If you want to exercise for longer than a few seconds, however, you're going to need to use other sources of fuel besides ATP and creatinine. This means you're going to need to use the other pathways in the body like glycolysis and the TCA cycle. These pathways are slower, and the result is that your exercise intensity diminishes with time. And you should know this if you've ever tried to maintain a full-out sprint. You can't do it for more than a few seconds, and that's because your ATP and creatinine decline, and therefore you have to rely on slower metabolic pathways to generate the fuel for your muscles. It's important to know that the calcium that's released inside of your myocytes when you exercise can stimulate metabolic pathways. In particular, it can activate specific enzymes so that glycogen breakdown occurs at a faster rate and so that the TCA cycle speeds up. And this is all in an effort to help the muscle cells get more energy quickly. This is a slide from the module on glycogen showing how glycogen breakdown is enhanced by calcium. When calcium is released inside of myocytes, the calcium calmodulin complex can stimulate glycogen phosphokinase A. This will phosphorylate glycogen phosphorylase, which makes the enzyme more active, and that speeds up glycogen breakdown, which is what you want to have happen when you are exercising. Here's a slide from our module on the TCA cycle, and what I want to show you here is that calcium can activate isocitrate dehydrogenase. It can also activate alpha-ketoglutarate dehydrogenase, and both of these effects will speed up the TCA cycle inside of exercising muscles. You probably know that there are two types of exercise, aerobic and anaerobic, but let's talk about what those two types of exercise mean on a biochemical level. Aerobic exercise includes activities like long-distance running. This is a coordinated effort by multiple different organ systems. Your liver and your fat cells and your muscles all work together to supply energy from many different sources to keep you going on a long run. Anaerobic exercise, on the other hand, includes activities like sprinting and weightlifting. This is purely a muscular effort. In fact, in many of these activities, the blood vessels in the muscles are compressed during peak contraction so that you don't get more blood flow to the muscles. The muscle cells are isolated from the body in that case, and the muscle has to rely on its own fuel sources. So let's talk about the biochemistry of anaerobic exercise first, and to do this, let's consider the case of a 40-yard sprint. If you were to do a 40-yard sprint, the ATP and creatine phosphate in your muscles would be consumed in the first few seconds. At this point, your muscle cells would try to rely on their own internal stores of glycogen. However, they would have to metabolize it to lactate. That's called anaerobic metabolism. And that's because the TCA cycle is too slow to generate enough glucose and enough energy for the muscle cells to use. As a result, the fast pace is not going to be able to be maintained for very long. The creatine phosphate will be consumed and lactic acid will accumulate in muscles. And you may know if you've ever tried to sprint that it's very hard to keep up that pace for more than a few seconds. Now let's talk about the biochemistry of aerobic exercise. And first, let's consider moderate aerobic exercise. And let's talk about what happens when you go on a 1-mile run. The ATP and creatine phosphate in your muscles is consumed in the first few seconds, as we talked about in the last slide. Your muscles will then begin to metabolize glycogen, but because you're not in an all-out sprint, they can use the slower metabolic pathways like the TCA cycle and electron transport. In other words, they can metabolize the glycogen all the way to carbon dioxide. This is aerobic metabolism using oxygen. As a result of waiting for these slower pathways, you have to run at a slower pace than in a sprint. But this is nice on your body because there's less lactate production and it allows time for the TCA cycle and oxidative phosphorylation to occur. And you may have heard of runners who do what's called carbohydrate loading. This means they eat lots of carbohydrates before a run. And what they're trying to do is increase the amount of muscle glycogen so that they have more glycogen available for providing fuel during a jog. Now let's consider the case of intense aerobic exercise. And to do this, let's consider running a marathon. When you run a marathon, there's cooperation in terms of biochemistry between your muscles, your liver, and your adipose tissue. Once again, ATP and creatine phosphate in muscles is consumed within seconds and doesn't last very long. Again, your muscle will metabolize glycogen all the way to carbon dioxide aerobically. In the case of a marathon, however, your liver is also going to assist by metabolizing glycogen and producing glucose, which it will release into the plasma, and that can be taken up by muscles to assist in production of energy. Often all of the glycogen in your body is consumed during a long race like a marathon. And once again, this is why runners often carbohydrate load before long races. Because all the glycogen gets consumed, there's some conversion to metabolism of fatty acids as well. Metabolism of fatty acids is a slower process. And as you know, the maximum speed of running a marathon is reduced compared to the maximum speed of running shorter distances. And elite runners condition their bodies and their muscles to be able to run on stored glycogen and fatty acids for long periods of time at a fast pace. The hormones in your body speed up fatty acid metabolism during intense aerobic exercise like a marathon. Glucagon and epinephrine inhibit the rate-limiting step in fatty acid synthesis. They inhibit the enzyme acetyl-CoA carboxylase. This ensures that fatty acid synthesis will not occur during a long race like a marathon. As a result of inhibiting this enzyme, levels of malonyl-CoA will fall, and malonyl-CoA is a powerful inhibitor of beta oxidation, so by lowering its level, you will speed up beta oxidation. Here's a slide from our fatty acid module on beta oxidation. Normally, malonyl-CoA inhibits this enzyme, CPT1, but in the setting of intense exercise, malonyl-CoA levels fall. This will speed up the activity of carnitine palmitoyl transferase 1, and it will allow more fatty acyl-CoA to enter the mitochondrial matrix so it can undergo beta oxidation. You should understand the biochemistry behind muscle cramps. When you exercise too intensely, you consume all of your NAD. In other words, you speed up all those metabolic pathways we've been discussing, and all of your NAD gets converted into NADH. Now, normally, electron transport can convert NADH back into NAD, but if you exercise too intensely, you exceed the capacity of the TCA cycle and electron transport, and all of your NAD is tied up in NADH. This is often referred to as an elevated NADH to NAD ratio. When you have an elevated NADH to NAD ratio, this favors the conversion of pyruvate to lactate. As a result, all that lactic acid makes the pH fall in muscles, and this leads to cramps. For this reason, distance runners condition so they have lots of mitochondria and their electron transport chain has lots of capacity in the hopes of preventing lactic acid buildup in their muscles. To make this more clear, here's a slide from our glycolysis module showing the final steps in that process. Recall that NAD is consumed in this step of glycolysis. It's converted into NADH. Now, normally that NADH can go to electron transport and be converted back into NAD. However, if the capacity of electron transport is exceeded, NADH levels will climb. In order to fix this problem, pyruvate will be converted to lactate. This converts the NADH back into NAD+, so it helps the cell by generating the NAD+ that it needs to keep glycolysis going. However, it hurts the cell because it increases the lactic acid level and can lower the pH. For the remainder of this module, we're going to talk about the biochemistry of starvation. In order to do that, we're first going to talk about the biochemistry of the fed state because starvation is basically the opposite of the fed state. So when you eat a big meal, glucose and amino acids are all absorbed into your blood, and lipids are packaged into chylomicrons, which enter the lymph and then go to your blood. As a result of all of this, you have an increase in insulin secretion. The beta cells of the pancreas secrete more insulin. They're stimulated by the high level of glucose in the blood and also by the parasympathetic nervous system. Insulin drives all the biochemistry of the fed state. Absence of insulin drives all the biochemistry of the starvation state. In the fed state, insulin promotes glycogen synthesis in the liver and in muscle cells. It also increases the rate of glycolysis and inhibits gluconeogenesis. In addition, it promotes the transfer of glucose into adipose tissue so it can be used to form triglycerides. It also promotes the uptake of amino acids by muscles, and it stimulates protein synthesis and inhibits breakdown. And all these things are fairly obvious if you think about what the body wants to do when it has excess energy around, which is what is the case when insulin levels are high. You don't need to know every enzyme that's affected by insulin, but there are a couple that are particularly high yield, and we'll review those now. Glucokinase is an enzyme found in the liver and in the pancreas. Its function is induced by insulin. Insulin can promote its transcription. The purpose of glucokinase is to phosphorylate glucose so that it becomes glucose 6-phosphate and is locked inside of liver and pancreas cells so that it can undergo further metabolism. The rate-limiting enzyme of glycolysis is phosphofructokinase 1, and the rate-limiting enzyme of gluconeogenesis is fructose 1,6-bisphosphatase 1. Insulin influences both of these enzymes via an intermediate called fructose 2,6-bisphosphate. This is a complicated process that I discuss in detail in the module on glycolysis, but what you always want to remember is that the level of fructose 2,6-bisphosphate inside of cells functions as an on-off switch for glycolysis. When the level is high, glycolysis is turned on, and when the level is low, there is less glycolysis. This favors gluconeogenesis, and all of these effects are influenced by insulin. This is a slide from our glycogen module showing you that insulin can activate the enzyme glycogen synthase so that glucose will be converted into glycogen. Conversely, when glucagon levels are high and insulin levels are low, the enzyme glycogen phosphorylase becomes active so the glycogen is broken down into glucose. The rate-limiting step in fatty acid synthesis is the conversion of acetyl-CoA to malonyl-CoA. The enzyme that catalyzes this step is called acetyl-CoA carboxylase, and this enzyme is activated by insulin so that you get more fatty acid synthesis when insulin levels are high. Conversely, when insulin levels are low and glucagon rises, this enzyme will become inhibited. So all that is the biochemistry of the fed state, but what happens when you begin fasting or you enter a starvation state? Well, the glucose levels in your plasma begin to fall a few hours after a meal. This leads to decreased insulin secretion, and this will trigger increased glucagon secretion. The most important effect of glucagon is to stimulate glycogen breakdown in the liver. The breakdown of glycogen into glucose and the secretion of glucose into the plasma is very important for maintaining glucose levels between meals. This is the dominant source of glucose when you're not eating. If you don't eat for 6 hours and you do a fingerstick blood sugar, you will find that your glucose level is normal at about 90 or 100. The reason it's normal is because your liver is breaking down glycogen to supply glucose into the plasma. If not for that, you would become hypoglycemic and you could have a seizure. Other effects of glucagon in general are to oppose insulin, so it will do the opposite of what insulin did. Glucagon will inhibit fatty acid synthesis, it will stimulate the release of fatty acids from adipose tissue, and it can also stimulate gluconeogenesis. And I talk about all these things in other modules on fatty acids and gluconeogenesis. The graph on this slide depicts the sources of glucose in the plasma as a function of hours after fasting. So when you begin a fast, you consume the ingested glucose from your last meal within a few hours after you start fasting. At that point, glycogen becomes the dominant source of plasma glucose. However, glycogen only lasts about 24 hours. So beyond 24 hours, if you don't eat any food, you begin to rely completely on gluconeogenesis. Gluconeogenesis synthesizes glucose using carbons from other structures, and those other structures include alanine and lactate and glycerol and odd-chain fatty acids, and we'll talk about all of these in the next few slides. This is a picture of Gandhi here, and if you know the history of India, you know that Gandhi used to go on weeks-long hunger strikes, and I like to think of him on the third or fourth week of his hunger strike and think about where all that glucose in his plasma was coming from. So a couple of key points about glucose sources. The first one is that the glycogen in your body is exhausted after about 24 hours. If you see a board question describing someone who's been fasting for 3 or 4 days, the glucose in their plasma is not coming from glycogen at that point in time. Key point number 2 is that glucose levels are maintained in fasting by many different sources. The glycogen is quickly depleted, and all these structures here help to provide glucose for the plasma. So alanine is one source of carbon for gluconeogenesis. This comes from the alanine cycle. Muscle cells break down proteins and generate ammonia. The ammonia is passed to alanine, which is secreted from the muscle cells and goes to the liver. In the liver, the ammonia is removed and enters the urea cycle, and the alanine is converted into pyruvate. Pyruvate can enter gluconeogenesis and become glucose, and then it can be secreted from the liver and go back to the muscles to supply the muscles with glucose. So the key point here is that peripheral tissue alanine coming from muscle cells, is being used to generate glucose in the plasma to maintain the plasma blood sugar level during a state of starvation. Lactate is another source of carbon for gluconeogenesis. This comes from the Cori cycle. In the Cori cycle, muscle and also red blood cells metabolize glucose into lactic acid. The lactic acid is released into the plasma and goes to the liver, where it's converted back into glucose and secreted into the plasma so that muscles and red blood cells can use the glucose. So once again, this is a depiction of how a structure like lactate donates its carbons to glucose in the blood sugar that maintains the blood sugar levels during starvation. You can also get carbons for glucose from gluconeogenesis from glycerol. Glycerol is generated when triglycerides are broken down. The fatty acids are removed, and this releases molecules of glycerol, which can go to the liver, where an enzyme called glycerol kinase converts the glycerol into glycerol 3-phosphate. Glycerol 3-phosphate can then be converted into dihydroxyacetone phosphate, which is an intermediate in gluconeogenesis, and this can then be converted back into glucose. Therefore, you are getting carbons for glucose from molecules of glycerol that came from triglycerides stored in your adipose tissue. Finally, odd-chain fatty acids can also be used as a source of carbons for glucose via gluconeogenesis. Normally, beta oxidation completely consumes fatty acids, but this is different in the case of odd-chain fatty acids that have an odd number of carbons, for example, 9 or 11 or 13. In the case of odd-chain fatty acids, beta oxidation stops when 3 carbons remain. The structure that will be left when 3 carbons remain is called propionyl-CoA, and this can be converted through a series of steps into succinyl-CoA. Succinyl-CoA, you may know, is an intermediate in the TCA cycle, and it can be used as a source of carbons for gluconeogenesis. And the key point here is that only odd-chain fatty acids can be converted into glucose via gluconeogenesis. That's because even-chain fatty acids are completely consumed via beta oxidation. So we've been talking about how gluconeogenesis maintains your plasma blood sugar level during fasting and starvation, but another reason that level is maintained is because glycolysis slows down from the low insulin levels. This means less glucose is used by the muscle and the liver. As a result, they shift over to fatty acid beta oxidation for fuels, and this also spares glucose so that when you're fasting for days or even weeks, your blood sugar level will remain normal and won't get dangerously low. We'll finish this module by talking about a couple of derangements of fasting and starvation biochemistry, and the first one we'll discuss is kwashiorkor. This is a form of malnutrition that occurs in impoverished countries where children or even adults have inadequate protein intake. As a result, they develop very low albumin levels, and this contributes to edema. So this picture here is a child with kwashiorkor. You can see that he's very skinny because he's malnourished, but in addition, his feet are puffy and so is his abdomen. And the reason those structures are puffy is because fluid is leaking out of the intravascular space because it can't be held there. There's not enough albumin present to hold it in the intravascular space. So children or adults who have kwashiorkor are very skinny, and they also have swollen legs and swollen abdomen, and the reason is because of inadequate protein intake. The other type of malnutrition is called marasmus. This is when there's inadequate energy intake, not enough intake of total calories. This is like kwashiorkor without the edema. There's muscle and fat wasting. Many people describe it as skin and bones, and this is a picture of a father and his son who are suffering from marasmus in an impoverished country. Finally, let's talk about hypoglycemia between meals. As I explained before, the body has a number of mechanisms to maintain the blood sugar level during times of fasting and starvation. But in children who have metabolic disorders, sometimes these metabolic pathways fail. One of the ways children can develop hypoglycemia between meals is if they have a glycogen storage disease. I talk about these in detail in the module on glycogen, but for our purposes here, just know that these disorders result in hypoglycemia and ketosis between meals. It's usually after an overnight fast because at this point in time, the children are heavily reliant on their glycogen stores, and if they have a storage disease, they won't be able to maintain the plasma blood sugar level. Another reason children can develop hypoglycemia is if they have a hereditary fructose intolerance. I talk about this disorder in the module on fructose and galactose. These children have a deficiency of an enzyme called aldolase B, and they build up fructose 1-phosphate, and this ties up all the phosphate in the form of this molecule, and they don't have enough phosphate to generate ATP. These children develop a number of symptoms, including hypoglycemia, and it usually happens in a baby who is just weaned from breast milk and begins consuming fructose. Hypoketotic hypoglycemia refers to low glucose and low levels of ketones, and this is very unusual, but it can occur in the setting of fasting if there is a problem with beta oxidation. So normally when you are fasting, free fatty acids undergo beta oxidation and become ketones, but if there's a deficiency in beta oxidation, then ketones will not get synthesized. Because there's a lack of ketones, tissues will overuse glucose, and this will lead to hypoglycemia, and the combination is called hypoketotic hypoglycemia. It is seen in beta oxidation disorders. There are a number of beta oxidation disorders, and I talk about some of these in the video on fatty acids, but two that you should know for step one are a carnitine deficiency and an MCAD deficiency. In a carnitine deficiency, the levels of serum carnitine and acylcarnitines are low. An MCAD deficiency is an enzyme deficiency syndrome where children are born deficient in the enzyme medium-chain acyl-CoA dehydrogenase. This leads to high acylcarnitine levels. In addition, a classic finding is dicarboxylic acid, 6 to 10 carbons in length in the urine. And I talk about both of these disorders in much greater detail in the video on fatty acid metabolism. And that concludes our video on exercise and starvation.